Yes, welcome to the day 7 session on types of force field, quantum mechanics, and de novo drug design. Okay, so this is actually day 7 session topics, and in day 8, on tomorrow we discuss on the ligand based approach like QSAR, QSPR, QSTR, pharmacophore modeling, etc. Okay, now let me take you to the recap of last session topics. So, can you mention about what exactly did we discuss in the last session? Good, very good. So, we discussed on protein-protein interaction databases. That is, we discussed on even extensively on protein annotation database, UniProt. UniProt refers to Universal Protein Data, Universal Protein Web Resource. In this web resource, we have plenty of databases like UniProt Knowledge Base, Proteo, Uniref, Uniprotein Archive. Like this, we have plenty of databases present in UniProt. The objective of we have gone on UniProt is actually it is a fully annotation database. It means that if you want to get more details of a protein up to the research, we can able to explore on UniProt. Okay, this is extensively mentioned for the proteins. This is a unique protein database. Then we have gone to the organism specific databases. Okay, we have come across various databases under organisms. Then we have gone across the annotated databases. Majorly, we discussed on UniProt for sequences. For structures, we have seen RCSP, PDB. Okay, and till now I didn't taken you to the MMTV molecular mechanics. I think 2 days before I have taken you to the NCBA. In the NCBA website URL, you have to go to the structure. Inside the structure, if you type a protein, whatever protein structure is going to take you is from molecular modeling database. Then we have discussed on various PPI databases, protein-protein interaction databases, we have seen StringDB, BioGrid, and then we have seen gene expression databases also. And then we discussed on a proteome database from UniProt, and then we have discussed on molecular modeling tools. So majorly we discussed on small molecular modeling, isn't it, on yesterday? So small molecules are generally chemical compounds, drugs, peptides, okay, even including RNA. For RNA, we can now go through the SIM-RNA server. Inside the SIM-RNA server, we have to provide the RNA sequence. To get the RNA sequence, we can go to the NCBI nucleotide. Under NCBI nucleotide, get the mRNA sequence, the RNA sequence, and then you can get the structure for that. Okay, and related to StringDB, so this is the largest protein-protein interaction database. We explored like how to download the PPI data set, okay, in the form of .tsv from the StringDB. And also we discussed like how to increase the interactions. Defaultly, we can see 10 interactions. If you want to increase it to the 100 interactions, how to make it using the settings. And whatever we download all the 100 interactions, how to open this in the cytoscape, how to visualize in the cytoscape visualizer. So this is what we have discussed, isn't it? How to open a protein file, protein, that is 3D structure of a PDB protein file in Discovery Studio Visualizer, and how to read the coordinates of the protein structure using Notepad. So this also we have discussed. And then on yesterday, exclusively we discussed on how to open even the data, whatever we downloaded in StringDB in Excel sheet. Okay, and how to visualize the interactions using visualizer. I told you plenty of apps available for the freshers. Initially go to the help manual as well as YouTube videos present in the help menu of the cytoscape. Okay, can install as many number of apps present in the cytoscape. It's easier to use, and it's of course an open source software. We have discussed all this information. Okay, and then anything left out, we discussed also extensively on DigiNet. So what are the different indices available in DigiNet? This is disease zone database. DigiNet is a popular disease zone database. So for a particular gene, okay, what are the types of diseases associated with it? So for actually related to disease zone database, even we can get this connection even from our impact database. You know, Indian medicinal database, traditional medicine database. We also have disease zone database. Okay, we have drug protein interaction as well as disease zone. So like this, we have discussed on the other kind of omics diseasome, which is now a contemporary kind of a research in database development. Okay, and we have also seen the computational development, how the computational databases are developed. I told you initially we can see the development of genome. Later came to the proteomic era. Now we are coming across interactomics. So this year, okay, this decade, we are many scientists are focusing on the RNA research. Okay, interactome kind of a research. They are extensively extending the length of the database of interactors. Okay, so this is what happening in the world. If you go to any country, US or European countries. So now this is what we discussed, isn't it? Can we go ahead with today's session? So before going to today's session, I can show you some hands-on, like something whatever we missed in earlier sessions. So if you see keg, okay, so also I got one kind of comment from a feedback. So I want to explore more on the pathway databases and especially keg. So today I'm explaining you from the cake. Okay, what are the databases present and how to search the data from the cake. Later we can connect with this seventh day session. So in cake, we have a cake pathway database. So because it's a kind interconnection of the yesterday session. So if you want to download any kind of a disease pathway, because this is most important. See, just knowing the database and knowing the data won't help you. One should able to designing the pipeline, like what are the sequence of events to be done for drug design, and what are the sequence of design methods we use for vaccine design. This is most important for research. Okay, so if you want to start the research, okay, if you initiate the research, okay, so the first thing what if you want to search on a particular target, okay, and if you want to get the information, I suggest you to go to the uniprot, get the target information. So whatever target information you can get for that is a protein annotation you can get. Later you have to search the information in the pathway. The largest pathway database, extensive pathway we can able to see in Kyoto Encyclopedia of Genes and Genomes from Japan website. So if I want to focus on disease research, okay, so we can go to the human diseases. So in the human diseases, we can see the types of cancer, okay, colorectal, pancreatic cancer. For each and every cancer also we have plenty of unique cancer databases. Okay, for colorectal cancer, we have more than 10 databases for downloading the data sets. And similarly, for liver cancer, we have plenty of cancer, public domain cancer databases also, other than NCBI, Uniprot, DDBJ, or Genome.jp, whatever we are discussing. Okay, so I'm also taking an extensive courses for the cancer research. If you want to step on cancer research, how to first initially do the in silico studies and how to design the in vitro and in vivo. So all this also I'll be conducting courses in future. I've already conducted for one company. Okay, now let me take you to this. Okay, at least I can give some basic knowledge on the cancer. See, there are different types of cancers available: infectious disease, viral, bacterial, parasitic, immune diseases. Okay, so also worked on the rheumatoid arthritis. For the rheumatoid arthritis, whatever I worked in computational part, only I tried with the computational part. But fortunately, I received two Young Scientist International Travel Grant Awards. one from CSCR and another from this ICMR. Actually, we have plenty of opportunities. Even the UG, PG, PhD scholars have plenty of opportunities to work on computational part, because working on in vitro studies and getting the results takes more time, and even it's more expensive. If you work on even a small part of the computational studies, you will be getting a chances to present papers in international conference with grants, sponsored with grants. Okay, so focus on even an outcome in the form of a research. At least complete a mini project or major project. Okay, and then you can, for if you want to work or the other colleges, if you want to work project under my guidance, you can able to work under consultancies. Okay, so the my college is anticipating to link the other college students in the form of consultancies. So that is something different. So you can contact me later. But now I'm giving you some idea on that. Okay. So just the theory and practical knowledge won't help you. Now I'm also giving you skills to how to connect with this pipeline. So I told you that I worked on also a small part of the rheumatoid arthritis where I got a grant. So how exactly I worked, So initially, if you see this rheumatoid arthritis, and also my PhD research majorly focused on dendritic cell. Okay, the dendritic cell is a professional antigen presenting cell, and this cell, if it interacts with the T cell, okay, this T cell is going to get activated, and one T cell is going to proliferate into two T cells. So the T cells and dendritic cells population, you know how the dendritic cells can multiply, even if any kind of a cytokine, if any kind of a ligand, if it interacts with the dendritic cell receptor, this can able to activate dendritic cell, and it can also get proliferated. So just because of these immune cells can be macrophages, dendritic cells, T cells, if it is present in the synovial fluid, no, arthritis is nothing but a kind of a pain in the joints. I believe we have around 26 joints, I believe. For the 26 joints, we have 26 26 factor. Like out of 26 joints, if a patient is suffering from 13 joints, then 13 by 26 is the factor. So the pain level is 50%. So this is how we measure. How we measure the blood pressure? We measure through systolic and diastolic pressure, isn't it? If you want to design machine learning algorithms, so one should know about the disease, and one should know about the parameters, what parameters we have to fix. Okay, and similarly here, if you see in this kind of an arthritis, so we can see the excess immune cells present in the synovial fluid in the joints can increase the pain, and it aggravate the arthritis. So what are the inhibitors we can able to take? If you see, so whatever cytokines generated, okay, the GMCSF stands for granulocyte macrophage colony stimulating factor. This is a growth factor used for growth of macrophages and also dendritic cells. If it GMCSF linked with IL-4 can generate dendritic cells instead of macrophages. So one thing you have to remember that these growth factors can also cause inflammation, synovial pannus formation. Okay, and I can't able to focus all these kind of target receptors for performing talking. So I just targeted one important receptor out of plenty of target receptors, whatever you find. For example, if you say this VEGF is responsible for angiogenesis, and these chemokines are responsible for inflammatory cell infiltration, joint destruction for the MMP. Okay, the molecular pathogenesis proteins are responsible for joint destruction. Like this, you can see plenty of targets. I just focused on one or two important targets, and then I talked and I checked whatever plant compounds I talked with these one or two targets, or talking with the appropriate binding sites of the target organ. So I just reported this. A small part of a research, I didn't even work on MD simulation, but that kind of a small research also received two types of grants. Okay, so this is what I want to mean. So just don't limit up to the target. You should mention about the pathways and you should mention about how this target protein can uh, can influence that kind of a pathways. What kind of symptoms we get with respect to that target, and if we target through the inhibitors, what is the ultimate effect? So all these things we have to describe clearly. Okay, so to the pathways, if you want to work on disease pathways, can be viral, bacterial, microbial, or autoimmune disease, cardiovascular, neurodegenerative. So most of the diseases, whatever you're working, okay, so the important diseases we can able to see here. If you don't find any particular disease, then obviously we have to go to the uniprot and we can go to the corresponding kind of a PPA interactions. Then you can try to see how what is, whether it's the positive regulation or negative regulation, and what is the extent of positive regulation, negative regulation. So all these things you can able to check from the p values, and you can able to mention about that. Okay, mention everything in your research. So this is how. Don't simply restrict up to the target and you can tell I have completed a project. You should show something about how it influences your target is linked with one pathway or multiple pathways. Got it? So, and also other than this, we also have metabolism pathway. Like if you are working on bioprocess, and if you are producing on, my startup majorly focus on secondary metabolite production. Secondary metabolite. What do you mean by secondary metabolite? Can someone tell me what are the examples of primary metabolites in plant? Primary metabolite in plant. Of course, primary metabolites default we mean in plants. That so I got answers immediately. Carbohydrates, proteins. That's it. Anything else? Only carbohydrates, proteins. That say a type of catalytic protein enzymes, fats. Very good. Fats, fatty acids, nucleic acids. So, superb. These are primary metabolites. Then what are secondary metabolites? I can show you now. So secondary metabolites, you know that, but I am showing you however. So you can see phenylpropanoid biosynthesis. See how a plant can synthesize the phenylpropanoid, stilbenoid, flavonoid. I work on flavonoid, flavones, steroidal lactones. So these are the molecules, but I am also focusing on other molecules also. Okay. So we also link with the plant tissue culture of IIT Madras and worked few things. Okay. So already linked with this kind of institute for completing my postdoctoral associate, and even my PhD scholar is doing extensive research in IIT Madras as we have generated the fund. Okay, so you can see you can also link with the central institutes to work on a lot. Okay, in drug and vaccine design. Okay, now let me take you to this. Okay, in isoflavonoid biosynthesis, so many metabolites. Okay, we have hydroxide. So see, if you see these are enzymes, plenty of metabolites, more than 100 metabolites and more than 100 types of enzymes are involved in a particular pathway. Okay, in the isoflavonoid biosynthesis pathway. So like this, okay, if you go for plant tissue culture, okay, this kind of pathway is required for us. So now let me take you to the what are the other databases we can able to find in the cake. So other than the the pathway, we can also search for the drug. For example, if I want to search for the drug like aspirin, so we get plenty of aspirin and its derivatives. Like however you search in the drug bank, however you search in the NCBA, we can also search in this cake drug. Well, one thing unique in cake, other than the drug, other than the pathway, the most important we can find is the cake enzyme. Okay, we have a separate database for enzyme. If you want to focus on a particular enzyme, okay, we can able to type any enzyme. So protease is overall thing. So like even in a like ATPase, you can type ATPase or whatever Okay, and you can find much information. See, plenty of enzyme commission numbers are mentioned. Okay, you can go to the specific enzyme commission number of the protease and you find the detailed description of that. Okay, and also you can see this is also linked with the Brenda database. This is the largest enzyme database. So you have Brenda database ID. So for this, you don't have even information in Brenda. Earlier, whatever I opened, we have a connection to link with the Brenda database. Yeah, yeah. Here also we have Brenda. So like this, or if you want to work on enzymes, obviously one has to refer to the cake, and which can also link with this largest enzyme database called Brenda. So like this, we can able to see much information in the cake database. Now let me take you to the today's session topic. So now whatever you ask me in the feedback, I have completed my session. Okay. So whatever you want any sessions to be exclusively taken, you have to mention me, okay, in the feedback or in the WhatsApp. Even we have a common WhatsApp group, isn't it? So that I can dedicate exclusively for. Okay, either you can chat in the Google Meet, or you can able to express even through messages in the form of private WhatsApp. So okay, now, let me take you to the chapter 7. The chapter 7 is on types of Forcefield quantum mechanics and de novo drug design. So, let me take you inside. So, molecular dynamics, quantum mechanics, force fields, energy minimization, probe charming, de novo drug design. Molecular dynamics. So, molecular dynamics is a computer simulation method for analyzing the physical movements of atoms and molecules. Okay, and of course, this is over a period of time. And for this molecular dynamics, of course, we are using the classical mechanics, and classical mechanics uses Newtonian's equation of motion. Other than that, we use statistical mechanics and quantum mechanics, okay, in the molecular dynamics, as I explained you on yesterday. So if you see in this a type of simulation called Monte Carlo, in Monte Carlo simulation, you see in a protein, okay, you have a, if you have a surface like this in a protein, and whatever blue dots you can find or amino acids of a protein, and generally the if this is your 21st position, whatever amino acid here you can find is the 21st position, can also be close to something like a 154th position. If 154 amino acid protein is there, 21 can also be very close to 154. How it can be? Based on a kind of a folding kinetics. You know how a protein fold is based on protein reaction engineering. So we have different types of kinetics, like however you studied in chemical engineering, zeroth order kinetics, first order, second order, third order. Similarly, in protein reaction engineering, we have these four order of kinetics. So based on the protein, we'll be finding that kind of a kinetics, and of course, the folding is based on that. And this can be measured even through Monte Carlo. Not only we can able to see the dynamics of a protein, but we can able to measure the dynamics of a ligand protein complex. If ligand is bound with a receptor, and if you want to check the kind of a binding, so I mean the binding stability, the binding stability can also be measured through this simulation techniques. Okay, so Monte Carlo is one of the technique, and if you see this conformational, that is types of MD simulations, the first thing what we discuss is the conformational ends sampling. Here, the application is up to the binding pocket detection. So, if you want to detect the pocket, okay, I mean, detect the binding sites of a protein, we can use this conformational sampling. And I told you, if you see the binding sites, we can classify the binding sites into two types. One is the hydrophilic, the other is the hydrophobic. Hydrophilic and the hydrophobic. Okay, so these are the two types of interactions we can able to see in binding site detection. Other than that, if you want to analyze the binding paths, okay, so even in these sites, whether it is binding with appropriate binding sites or not. And if you see the catalytic proteins, that is enzymes. So for the enzymes, whatever we call as a binding sites, we can also call that as active sites. The term active sites is for the catalytic proteins like enzymes. Don't confuse if I tell active sites in between and binding sites. So binding sites are generally the regions where, if you focus on a disease, okay, so the disease, for the disease you have a specific binding sites. of a target. And if your drug or inhibitor, whatever we treat, if it can able to treat exactly, if it can able to bind exactly on the disease binding sites, then obviously we can use less dosage or number of dosages we can reduce. If that is protein binding, and also that specific targeting is not much precise, then obviously we are increasing everything. Okay. And if you see this kind of analysis, we can able to do with the conformational sampling rearrangement processes related to the detection of structural changes associated with induced fit or shifts in the relative population of conformations by ligand binding. More about the induced fit theory in molecular talking we'll be discussing on day 9 session. Okay, so just you can remember this is a part of talking. The rearrangement process, that is, if you see a protein molecule, okay, whatever we take, a target, whatever we take, we nowadays whatever visualizer we use, not whatever talking and simulation softwares we use, we make the protein as flexible. If you make a protein as flexible, okay, so the protein can have the functional groups, the protein can have functional groups with various conformations, and even the ligand can bind with the protein amino acids in various conformations. More about this we'll be discussing on top. Okay, so that kind of rearrangement process also we study in conformational sampling. So I told you the difference between talking and simulation. To recollect that, in talking, we simply measure the affinity, binding affinity of ligand and receptor. Okay, where a ligand receptor complex is formed. A particular concentration of a ligand, we react with the receptor. Okay, and if this is your forward rate reaction constant, and this is your backward reaction constant. So whatever ligand receptor complex we form, this is a product, isn't it? So we can, whatever binding affinity alone you study, okay, in terms of binding energy, we call it as torque. But in simulation, we also check with respect to over a period of time in the form of 100 nanoseconds, 200 nanoseconds, sometimes even we work on picoseconds. So all these things comes under MD simulation. And the next type of MD simulation, information derived from MD simulation, is energetics. That is a simulation of binding, unbinding, everything we can see in the form of thermodynamics. Of course, I told you in talking, whatever binding energy we calculate is nothing but change in Gibbs-free energy. The change in Gibbs-free energy is nothing but the change in entropy to the difference of product of temperature and change in entropy. And this is usually measured in the software through kilocalorie per mole. So this is the unit for change in Gibbs-free energy. That's what we measure as binding energy. Okay, in autodoc group. Can be any two: autodoc PVRX, autodoc 4.0, covalent autodoc. Okay, so in all the types of autodoc, we find these kind of calculation. And similarly, if you see, the next thing is the kinetics. I told you the kinetics is also a most important part. Not just the thermodynamics can play an important role in binding, but also kinetics, especially in the folding, in checking the folding parameters of the protein, and even how the ligand, if interacts with the receptor, ligand receptor complex kind of a kinetics. Okay, so we have types of kinetics to be measured. In terms of binding, also we can measure binding and unbinding. Also, we can measure the kinetics, not only thermodynamics, kinetics. Okay, so the kinetics is what I mentioned here. The rate of forward reaction is K1, and the reverse reaction is K minus 1. So based on the pH, based on a particular ionic concentration, or how exactly we are using, based on that only you can able to check whether you are forming a ligand receptor complex, or the ligand receptor complex is forming back again the ligand and receptor. You got it? This is a reversible reaction. Ligand and receptor, if it reacts, it forms a ligand receptor, isn't it? So this is a reversible reaction. So K1 and K minus 1 is based on the forward and backward reaction constants. Now let me take it to the quantum mechanics. So what is quantum mechanics? So till now we have seen the applications and the types of kind of an MD simulation. So but generally this quantum mechanics is a branch of science. Okay, is a branch of physics. We try to use an MD simulation. Okay, so we are discussing on that. So if you see the molecular mechanics, we classify it into three types. Once again, I'm recalling you. Number one is classical, number two is statistical, number three is quantum. Okay, so if you have many atoms, we study statistical mechanics. If you are studying a single atom with respect to Newtonian law of motion, we study classical. So generally, if we study classical, then we study statistical mechanics. If after the statistical, we go with the quantum. So this is the sequence we use. In classical, we study a single atom like Newton law of motion, F equals ma. Okay, and if you see if your protein is nearly 154 amino acid, and 154 amino acid has 1,000 atoms. For all the 1,000 atoms, we calculate force. So whatever statistics we work, and if you work on MD simulation, we work on energy trajectories. We work on trajectories for each and every atom. So if you have 1,000 atoms, you have 1,000 energy trajectories. So the statistics we use in statistical mechanics with the classical mechanics. In quantum, other than classical statistical, we also study the effect of photons. What is quantum actually? Quantum is nothing but something related to photons, isn't it? If a photon is going to hit an atom, okay, then obviously it's going to hit an electron, and you can able to measure it. You can measure it through the wave motion, okay, through the wave motion, and we can able to check the values. For simply understanding the quantum mechanics, I give you an example. Like if you want to measure the quantity of your protein, if you want to measure the quantity of a protein in wet lab technique, what exactly we can use? Am I audible? Yes. Am I audible?
Yes, sir.
Very good
Yes, sir.
interaction from participants. So yeah, in between whatever I'm asking, you're typing the answers. Excellent. So now I just asked you an answer like, what are the wet lab techniques we can use in our laboratory to measure the absorbance followed by weight of a protein? Either give the methods or equipment names, whatever you know. Bradford test, okay, you're mentioning up to that. But how do you measure that? Obviously, Bradford ultimate equipments. What equipments you use? Instruments you use. Good, superb. Colorimeter, UV spectrophotometer, electrophoresis. So think about this colorimeter, spectrometer, fluorimeter, nephlometer. In all the cases, we are using light. We are using light or not? Remember, in spectrometer also we are using a kind of a light, isn't it? So colorimeter also we are using light. So whatever light we use, okay, it's going to pass in the sample, and from that, even if it is having a more turbidity in protein, we can use turbidometer instead of spectrometer, colorimeter. We can use turbidometer. If you're working on amino acids, the amino acids have can show color. So for the color, we can use colorimeter. If your amino acids can also exhibit fluorescence, we can use fluorimeter. So in all the cases, obviously, we are using light, and just because we are using a light for measuring the properties of a protein through the molecules, how will we measure it? Only through the molecules. How many molecules are taking the light? Okay, that's most important, isn't it? So ultimate thing, whatever we work in our wet lab studies, we are utilizing the quantum mechanics. So according to the quantum mechanics, okay, it differs from classical mechanics in that we use the energy, momentum, angular momentum, and other quantities of bound system or restricted to discrete values. Generally, we measure. We won't consider atom as a single particle. We also use that as a wave particle duality. Okay, and we also use Hindenburg uncertainty principle to measure on this. The best example you can see here. Okay, if this is a single atom in a protein, single atom in an amino acid. Okay, this atom, okay, I mean this red color atom is moving from uh, here to here. So this is the length. So it's moving from here to here. Okay, here you can see the next thing. Okay, the same thing like an atom attached to the thing. If you just pull with high force, if you pull with high force, it is extending with more length. So based on how much force you give, based on the light, that is, if a photon can hit, then it can give a good kind of a spring-like motion. Based on the spring-like motion, you can able to see the wave-like kind of patterns are exhibited, and through these wave-like patterns, you can see the absorbance value increases or decreases. So how you can have a restricted rotation. This kind of a restricted rotation you can have if you have amino acids like cysteine, especially proline. Proline can have restricted rotation. So proline you have only one angle rotation. I believe 5 angle rotation, that to a restricted rotation. But if you have a glycine, the glycine can have all the 3 angle rotations. So generally, if an amino acid hits, then obviously can give these kind of a more flexible kind of wave-like particle motion. Okay, even though it moves like a spring, if you measure it, you will measure in the form of an analog signals. The analog signals is what converted as digital signals in the form of absorbance in these kind of measuring instruments. Got it? This is something about quantum mechanics. Now let me take it to the force field. In the context of chemistry and molecular mechanics modeling, a force field is a computational method that is used to estimate the forces between atoms within molecules and also between molecules. Okay, so you got it. So whatever I explained earlier, if you have a protein of 154 amino acids and all the each and every amino acids, you have multiple atoms, 1,000 atoms. Each and every atom you can measuring in the form of an energy trajectory. We sum up all the forces to get the total force. We sum up all the forces of all the 1,000 atoms to get the total force, and from the force we can calculate energy. The energy can also be a simple relation like force into distance. How much distance an atom can move, and what is the force we have given. Got it? So simple things. So to calculate the energy and to minimize the energy, we can use this kind of force fields. Force fields, whatever I mentioned, it's a part of a molecular mechanics, or we also call it as molecular dynamics. Also, okay, I can ask you one thing. What is the difference between molecular mechanics and molecular dynamics? Can someone tell me the difference between molecular mechanics and molecular dynamics? What do you understand from that? Molecular mechanics and molecular dynamics. See, this is actually a dry subject, but one should understand this. Just like that, studying the practical skills won't help you to write research papers. If you want to write research papers, one should have a knowledge on what exactly you're doing up to the atomic level. I asked a question like difference between quantum, what is that? Molecular mechanics and molecular dynamics.
Sir, molecular mechanics calculates the energy of a molecular structure, whereas dynamics is like just motion of atoms over time.
Superb, superb. Your answer is perfect. Uh, Still, I can give somewhat whatever you have missed. In molecular mechanics, whatever you mentioned is exact. In molecular mechanics, you can classify it as molecular statistics and molecular dynamics. Molecular mechanics can be classified as statics and dynamics. Generally, the molecule won't be in rest, isn't it? Generally, the molecule always have a kind of a motion. So, in molecular mechanics, whatever we study is in the form of a molecular dynamics in default, isn't it? Even if you pack gaseous molecule in the cylinder, it's going to exhibit the Brownian motion. And even if you try to dock a ligand with a protein receptor, it also has a kind of a dynamics. The ligand is not going to bind permanently with the receptor. Over a period of time, it has to wash away. Oh, how much it is going to bind, there's that much it's going to bind. That too over a period of time. So obviously, whatever we study as a molecular mechanics, nothing but molecular dynamics. Okay. So under your explanation is also uh, perfect. Okay, on the answer with respect to the answers. Now let me take you further. All atom force fields provide parameters for every type of atom in system. Okay. Now let me take you to the types of force fields. So classical force field, polarizable force field, reactive, coarse grained, machine learning, water, modified amino acids, and also we have other force fields. So whatever we discussed on yesterday, so we have two types of interactions: bonded interactions, non-bonded interactions. Sum of bonded interaction energy and non-bonded interaction energy, sum of bonded interaction energy and sum of non-bonded interaction energy can give you the total energy. This is what we have discussed on yesterday, isn't it? Of course, whatever we discussed about this energy calculation. Okay, this is applicable for molecular docking, simulation, as well as to check the folding stability of a protein. Okay, so for all this we can use this common concept. Now let me take to the classical force field. From the name itself, it clearly implies classical. It means that we are using Newtonian law of motion to calculate the forces. So the examples provided are Ampere, assisted model building and energy refinement. This is widely used for proteins and DNA. This is what most important. What force field for what application? And the second, what we discuss is consistent force field used for organic compounds. If you are working on chemistry, if you are a chemistry person, okay, you are synthesizing the chemical molecules, and if you want to check the stability, even the chemical molecules, whatever we find, we also see whether it's a form of a crystal or is an amorphous solid. If it is a crystal, we check on crystal lattices. We work on types of lattices. So many things are there, isn't it? Even in the chemistry. So those who are working on chemistry, okay, in the form of synthesis of organic compounds or polymers, metals. Even recent Nobel Prize winner got organic compound embedded with metal biomaterials. Okay, so all these things. Okay, one should have a knowledge on the consistent force field. Okay, see the applications and the charm. Many of the tools we use this energy information force field called charm. It means that chemist at Harvard Molecular Mechanics. So this is used for both small molecules and macromolecules. So only we can see in many of the tools we can have this kind of force field. And CVFF, consistent valence force field. Okay, also used for small molecules and macromolecules. Chromos, Groningen molecular simulation. And most of the force fields, whatever I mentioned, are commercial. For commercial, we have to pay and use, but we have also freewares. So wherever we have freewares, we can try to utilize it. So interface for force field, molecular force field. So these are the other force fields present in classical force field. The next is the polarizable force field. In polarizable, the name itself implies if you are focusing on polar compounds, or even your protein has more polar amino acids, okay, either this molecule or that molecule, okay, based on polarity. If polarity is measure of importance, then we can use these kind of force fields. See, the charm is coming under both classical as well as polarizable. Some can have a common kind of linking. And we have amber. This is also a common thing, isn't it? Amber we also seen in classical. Classical is still default. Without classical, we can't go ahead. But along with the classical, you can see even polarized. It also exists in polarized. So most of the force fields are classical, no doubt in that. Okay, but in polarizable, we have amoeba, polarizable force field, phased and oriented. The third is a reactive force field. So reactive force field, if you are focusing on enzyme and substrate. For example, if your protein is your enzyme And if a ligand is your substrate, got it? Ligand is substrate. This is what we find in enzymology. If an enzyme and substrate reacts, okay, it's not a, irreversible reaction. So I just use an eraser. So it is a reversible reaction, isn't it? If it is a reversible reaction, okay, so what exactly can find? Enzyme substrate is going to form, and this enzyme substrate can able to release enzyme and product. This is the final thing you can get. If this is K1 for a forward and K minus 1 for the reverse, and this might be a K2. This is about the kinetics. So if you find a kind of a reaction happening in the ligand receptor complex, then obviously one can able to go with the reactive force field. The example I've given as a EVB reacts FF. Next, coarse grained force field. If your drug is in the form of coarse grains, then better we can go with the dissipative particle dynamics, Martini, Sierra, BAM. Now about energy minimization. Till now we discussed is about the force fields. Now energy minimization. So whatever force field you use, we try to use to calculate energy. To calculate energy, we can also use this simple relation, energy equals force to distance. Later, we go with energy minimization protocol. Energy minimization can also be done using Discovery Studio Visualizer. You remember? Yesterday also I have showed you in the Discovery Studio Visualizer. So from BioVia, this is a product of BioVia. If you want to do animation, I can work with that kind of a tool also. Okay, energy minimization, also called as energy optimization, geometry minimization, geometry optimization, is the process of finding an arrangement in space of a collection of atoms where, according to some computational model of chemical bonding, the net intraatomic force on each atom is acceptable close to zero, and the position on the potential energy surface is the stationary point. So there are two types. Number one is steepest descent. Number two is conjugate gradient. Steepest descent, also known as gradient descent, is also known as gradient descent, which is a first order optimization algorithm. So this is used to find the local minimum or maximum of a function to take steps proportional to the negative or positive of the gradient. If this is steepest descent, conjugate gradient is another algorithm where you focus on a numerical solution of particular systems. Sorry, of a particular systems of linear equations, namely those whose matrix is positive semi-definite. Okay, so these are the two algorithms we can find in energy minimization. So energy minimization protocol is used for making the protein more stable. Similarly, if you want to make a more stable conformation of a ligand, we can go with the energy minimization protocol. So machine learning. So we can also use machine learning techniques for calculation of force fields. That is for calculation of force field, I mean even to calculate energy and followed by energy minimization. So for this, we can use F-flux. Okay, this is about machine learning technique and Probit charm. Now we can come to a new concept. Till now, we have discussed on what we mean by molecular mechanics, and we have seen the types of molecular mechanics. And then we have discussed on quantum mechanics specifically, that even we can use Schrodinger wave equation for estimation of energy, and also to measure about the quantum principles. Then we come across the types of force fields and its applications. Okay, and now we come to the Probit chart. What is this Probit charm? This Probit charming is a tool. Okay, this is used to detect structurally similar protein binding sites and also prediction of ligands from similar binding sites. So for this, I can use this Probit charming. So charming, you got it. This is the energy minimization protocol we have already seen. Okay, so this we have, so we can use it for various applications as I earlier mentioned. Okay, one such application is this prediction of ligands from similar binding sites and also detection of similar protein binding sites. Can we go to the Probis charming tool? I mean, this is an online server. So if you just type as a Probis in Google, it directs you to this. Simply have to search a protein through PDB ID. Type a PDB ID or you just give the PDB ID here. You must mention the chain name. So the chain can be A, or if you have multiple chains like A, comma B, comma C, if you are working on A chain alone, give that A alone. But if you want to work with all the chains, A, comma B, comma C, this is how we have to feed an input. Or even you can upload a PDB file. So simply uploading or providing an input of a PDB file, and also this is an optional for you if you want to receive, because you won't get immediately, but also you can better provide a kind of an email. So after uploading a kind of a protein file, if you upload a protein file, it takes some time to generate. But if there is already a PDB ID present in this protease database, you get a result instant. So in probe charming, okay, so after uploading a protein molecule, I can see series of compounds, a series of compounds, okay, which is specifically binding with this protein. So again, if you come across how it looks like, this concept is similar to the target deconvolution, isn't it? This also uses the target deconvolution, like just providing just a receptor. You get a series of ligands. So this is the best ligand, which is more specific. What is that ligand? N421 something. So just upload a protein molecule. The protein molecule I uploaded is 4WO5. This is the PDB ID I have uploaded, and the chain A. Okay, for this you can see the ligand, okay, as well as the PDB file. Okay, you can find around 395 similar structure, 395 PDB files. Whatever protein we have uploaded, okay, so we are getting a binding kind of an compounds, and or also we are getting a similar kind of a binding sets of the protein. And also you can see the types of binding sites, binding site 1, 2, 3. A protein can have n number of binding sites based on the surface, based on the Monte Carlo simulation. Okay, so based on the surface, if you have nearly 12 hydrophilic sites and 2 hydrophobic sites, 12 plus 2. There are 14 binding sites of a protein. For all the 14, we will be getting this information. So this is about Proby's charming. And now we can go to a new concept called de novo drug design. De novo, what do you mean by de novo drug design? So in de novo drug design, we can classify as atom-based construction and fragment-based construction. This can further be classified as structure-based and ligand-based. And if you see these types of design, so tomorrow we'll be discussing on ligand-based drug design in detail, QSCR, pharmacophore modeling. Okay, but all this also comes under the and structure-based talking and simulation. Now let me take to the de novo. In de novo drug design is a computational technique wherein novel molecular structures with desired pharmacological properties. Okay, so if you want to know more about the de novo drug design, I can show you this picture and illustrate. So de novo drug design can be de novo linking and de novo evolution. What is the difference between de novo linking and de novo evolution? See the pictures once. Now, can I explain you? Do you get something? See, if you have a fragment 1, 2, 3, think like you have 3 different fragments. Okay, and if this is your protein, and just because they mentioned active site, this might be an enzyme. Okay, so if you have a protein, okay, and if you have 3 drugs, okay, all these 3 drugs are binding with 3 different binding sites like A, B, C. So all these binding sites have different conformations. This conformation is different, and this conformation is different. So you can't able to bind a drug to the binding site A. So whatever the fragment of drugs we have, okay, it is binding with a specific binding site regions. It's binding with a specific binding site regions. The best example you can also consider in the form of an antibody. That you know the antibody. The antibody has 2 arms. Okay, so there you have just 2 arms, but still you have uh, 4 different regions. Okay, So based on this kind of surface, also we can get two different regions, or even based on the sites, we can have as many number of regions, SCFE fragments. Okay, even in antibody engineering, we should have some knowledge on this. Okay, with respect to specificity, how your antibody, the engineered antibody, can specifically bind with the binding sites. That's what antibody engineering. In biotechnology, we are progressing towards antibody engineering for precision medicine development, isn't it? For the types of diseases like cancer. For cancer and rare diseases, of course, we are working on engineering the antibodies. So for this, one should have these kind of knowledge. First, we see the simple thing. So whatever drugs fragments we have, okay, which is binding the specific binding sites, we can link with linkers. This can be linker one and linker two, so that it will be in the form of a single drug. And whatever we have designed, we can use it, okay, for the treatment. As in the evaluation, it won't be like that. So like however we have first initially fit the three kind of fragments in the three binding sites. Here we first try one by one. So first we have a major drug fragment like a two. Later we come up with one. So how this can be, again this can be linked with a linker. Generally, if you see this kind of a concept, we can also see in siderophore development. Siderophore. You know what you mean by siderophore? Attaching iron with drug. You know, especially for the bacterial fever, if the patients are infected with pseudomonas, especially pseudomonas aeruginosa. For the pseudomonas aeruginosa, you know, this kind of a pathogen majorly consumes iron in our blood. You know, in iron, we in the blood hemoglobin, we have complex iron molecule, the form of porphyrin structure, isn't it? So this iron molecule in the blood gradually is going to reduce because of this infection, and this infection majorly happens in lung. So this kind of a pathogen majorly affects the lung, and for this kind of a target, okay, whatever the if you want to design a drug, okay, if we use this kind of an ion chelator, we call this as an ion chelator. That is, first what we do is we link an ion molecule with a drug which can target specifically. If this is your drug and this pink color fragment is an iron molecule, okay, this iron molecule, whatever is going to attach with it, okay, it's going inside, and the Pseudomonas aeruginosa, if is it says the iron molecule, it immediately try to grasp, and if it grab, obviously it's also going to intake the drug. This is like a Trojan horse strategy. So whatever drug we give, we deceive the kind of a pathogen for the treatment. So deciding such a kind of a siderophore, and in some cases in the development of prodrugs, you remember about the prodrugs, if you want to release. So for this also we can use this kind of a strategy like a de novo drug design. That is something different. Okay, I'm not fitting with this. That is a different kind of a concept. The prodrugs can also be used in this kind of a de novo drug design. Now let me take you to the kind of a process how it happens. Target binding site. First we have to identify the target binding site, then we have to hit. Just I need 2 to 3 minutes to complete this today's session. So hit identification, and then we have to find the hit to lead. So these are the general process. So growing, linking, merging. Growing with a single fragment, and then we have one and two, and we are merging through the linker. We can also use linker, and some cases we can also use spacers. Linkers and spacers are used to link multiple fragments, and these are the steps in methods in fragment-based drug design: growing, linking, and merging. So this is something about de novo drug design. Okay, we can. There's a lot to know more about the theory, so you can check in internet. Okay, so at least I've given some sufficient knowledge on the de novo drug design. Now let me take you to the Probase server. So in Probase server, we can work on one particular protein molecule. what molecule I tried there, can I work the same molecule? Or if you know any PDP, you can mention me. So the molecule I have tried is 4WO5. This might be a sample molecule, I don't know. Whatever I tried, you can try with your own molecule. Okay. So 4WO5. And this is the chain. And if I want to give a kind of an email ID, give an email ID, submit job. So enqueued, okay, so it's getting loaded. So if the results are getting generated, so you can able to see the results similarly like this kind of a results you can able to see after this. Okay, so it takes some time. So just 1% is completed. So tomorrow I can show you the result. If I receive it in, I'll be receiving it in email ID, isn't it? So tomorrow I can show you this kind of a result. So today I have completed my session. So I especially thank all the participants because even you joined on the holiday. So I appreciate your kind of an interest, okay, your enthusiasm in learning the skills in this course. Any doubts, you are welcome. I've completed my session today. Any doubts? No? Okay. I think even I don't find any kind of questions. Once more, explain about energy innovation. Energy innovation to make a protein more stable, we go with energy minimization where your calculated energy will be reduced. Initially, you can get more energy, but after energy minimization, the energy will be reduced. It means that you make more stable conformation of your protein. Okay, so that's what energy minimization. What is function of ion chlorine binding? Ions, this is what you got now fragment based activation, isn't it? So these ions can bind the specific sites that can also give some kind of function. Especially just now I was explaining you about Pseudomonas aerogenes, how to target Pseudomonas aerogenes. So Siderophore, working on Siderophore is the best strategy to work on because treating the lungs infection is very very difficult when compared to any other organs because we have so many alveoli. Okay, so treating the lung infection is difficult. This can be made. You can make a strategy that is this kind of a Trojan horse strategy to receive it. So here you can see how the ions can bind and how the ions can linked with a drug to do that. Okay. Is there any books for knowing more? We have plenty of books. I studied more than 20 to 30 books and I am giving this information. There are plenty of textbooks available. And nowadays, when you're choosing the chat GPT, perplexity, use this also. There also get so many references, journal paper references. That can also help you. So I suggest you use Molecular Modeling by Anand Solomon. If you want a specific book, the best book I prefer you is to study. He also called as a father of bioinformatics in India, I believe. I don't know. When I'm studying, they call him like that. Molecular Modeling by Anand Solomon. Okay, a small book, very easy to learn. So thank you very much for your patience and interaction. Have a great day to you all. Bye.